Thank you. It's my pleasure to be here to present my presentation here. My name is Dae-Sik. It's in Promontory. ETRI is one of the largest government funding research institute in Korea. Is specialized in the information technology and the fusion technology and AI technologies. Yeah. I'll talk today is the lung cancer sensors. It's a commercial biomaterial, and I'm not a sensor person, so sensors and early screening. And I applied the sensor to the early screening of lung cancer with the help of Seoul National University Hospital. My background is I am doing research on the biomarkers-based healthcare devices. The first, the biological, and it's based on the protein and the gene and the cell-based ones, and another one is chemical ones. Is organic compound. It's organic compound is more smaller than the protein, and this it's very small molecules. I want to combine the multi omics and connect to the our mobile device and doing to digital scale applications. Especially our team is research scope is usually we are developing the basic technology and making prototype devices like ones and lab-made ones, and we technology transfer to the company and technical technology investment to the through the ETRI Holdings is one of the investment ETRI owned companies for the investment. So I have some experience to the technology transfer to several more than 10 times. So among them, I have fortunately I am founding two companies. One is PCR companies is what it was registered in the Costa, like in the Nasdaq in US. Another one is to the immune device testing kit. It is the Sujantech. It is an immune testing kit device companies, and it is also registered in the Costa in Korea. It under FDA approvals and during the COVID-19 period. So I want to expanding to this the gene-based one and this protein-based one, and to the brioche organic compound. It's to the nose smelling. It's electric nose systems. So we did the technology transfer to the companies, and we are today. I want to do some result from at this part. Basically, breath is very important because no breath is dead. And so it can give the information about the lung cancer and the stomach cancers. So it's basically the volatile organic compound is analysis to the analytic chemistry instrument like GC-MS, PCR-MS, and immunologically it's antibody-antigen reactions. But usually using the sensors like human no, it can detect the VOCs using the sensor array and then analyzing with the AI machine learning, and they can classify the lung cancer or healthy persons. You know, lung cancer is the second most popular cancers among the all types of cancers, and the most lethal of cancers. It the most difficult for the lung cancer. It is a detector in the early stages. Usually, you have pain and then go to hospital more the page 3 or 4. So it is very important to early detection for the proper treatment at the early stages and then reduce the mortality rate. Recently, the breast cancer diagnosis disease is one of the interesting and designated one of top emerging technology in 2021 in this World Economic Forums, and that the market is increasing rapidly. The previous report is I selected ones. It is published in 2017, and that used 14 sensors and using the LDA, fuzzy neural network machine learning that shows the performance and accuracy and sensitivity and selectivity more than 90%. So it is a very prototypes. So we also did this research for over the 20 years. We tried in the National University. We made these devices and we applied to the early screening of lung cancer patient and in the National University hospitals. And at that time, we using the multi-layer perceptual neural network and 7 semiconductor metal oxide sensors, and we got the 75% accuracy. So we want to more to research on this improving the performance. So we made another instrument device like ones, and using the machine learning, so we applied to the early screening of lung cancers. You are focusing on the developing sensors. Sensors, many nanotechnology based ones, metal oxide sensors, carbon nanowire sensors, and the peptide biosensors, and the metal organic calorie battery sensor, and the bacteriophage sensors. We put together the different working mechanisms, and then put together the signals, and we apply the signal to the machine learning algorithms like convolutional neural network and virtual perceptron, and we can improve the performance of sensitivity and this specificity. The biomarker is very important also in these areas, and where they are well-known protein biomarker and genetic biomarkers. In this case of VOCs, there are several VOCs of specific cancers, lung cancer. In case of lung cancer, toluene, lidocaine, and trimethylhexane, and this kind of VOC was unique. existence in the lung cancer patient breath. And in case of diabetes patient, the acetone is one of the most well-known biomarkers. So it is one of our ongoing research topics. And we employ the multimodal sensing concept. At first, we usually semiconductor metal oxide sensor electric signal detection, but then in this time we applied it to the electrochemical sensors and the photonization detector sensor. So we signal from the 10 with semiconductor metal oxide sensor, one Ph.D. sensor, and electrochemical sensor, and to put together to make this pattern and to apply to the machine learning algorithm to get the classification of the disease. So this table shows for this clinical testing, we employed the commercialized sensors. We also basically sense nanomaterial based the sensors, but and for the clinical testing is the reliability and the sensitivity is very important property. So we are using for the clinical testing and with the commercial sensors. We got the multimodal sensors through the multimodal sensor is the sensing signals and the clinical data collection is and then you apply it to the AI algorithm for the with the data pre-processing and the deep neural networking, and we apply the three kind of machine learning algorithms: convolutional neural network, and then multi-layer perceptron, and recurrent neural networks. So we did basically simulation through the some tools and made electric loads electric signal processing part, and then the PCB design and fabrication and the flow controlling systems like this. And we gathering the breath sampling with the Telecommunications. It's from the patient in the hospitals. Yeah. Before the clinical testing, we check the basic sensor for response pattern test. Even actually, in the human breast is more than several hundred kinds of VOCs existed under the level of ppm, part per billions, and so we check the one ppm levels. VOCs, isobutyren, and formaldehyde, and ethanol, and benzene, and it shows the sensor is it's in this case 11 sensors, and it shows a different pattern according to the VOC. So this pattern is like a fingerprint tool. It can be biomarkers. We designed the clinical test set with the help of the surgery doctors and so National University hospitals. We employed the lung cancer patient 107, Dae-Sik control 74. We before the for the clinical testing, the doctors doing surgery in the morning. And before the surgery operation, we got the breast sample from the patient, and then And then during the surgery, cut the tissues, lung cancer tissues, and we check the stage from the 1 to 3, 4. We are targeting the early diagnosis of lung cancer. So most of more than 70% is stage 1 and 2. So we got the patents, sensor patents from in case of the semiconductor metal oxide, it has to the health control, it's to the Ph.D. and the chemical ones. And this part of the lung cancer pattern it shows different patterns and sensor responses. So we applied these patterns to the machine learning. The clinical data set, as I mentioned before, is 74 health control samples, 107 lung cancer samples, and before the learnings, we normalized the sensor response data like this, and we got the data 19 sensor arrays and 2,000 to 1,400 second response maps like once, and then we use one convolutional neural network and the 3 dense layers, and applying Lickey rule activation function and the batch normalizations. The classification, the lung cancer and the health control, right? For the classification. It shows that actually we are 90% was for the training, 10% for the test. We got the improvement, healthy control, personalized, and pathological data validation. Nearly among the 74, 71 one is healthy, and for the lung cancer 107, and actually lung cancer and to the lung cancer 105. The sensitivity and the specificity is more than 95%. So a very surprising result. It is more than is similar or is more than PCR result. So we are very excited with the result. We further studied for the we suggested the multimodal is better than the single motor sensors. Just applying to the semiconductor metal oxide, the sensors in a race, the accuracy of the for the training is 96%, for the testing 85%. But we applying the multimodal algorithm, at the training, the result was 99%, and to the testing data, 92%. So it's multimodal is better than single modals sensing. We are also checking the comparison classification performance in the different deep learning models, MLP and the recurrent neural network and the convolutional neural network. They show now we can what learning algorithm we choose the result the different. So among them, the 3 different learning module, so CNN is the best. So proper selection of neural network modeling and strategy, multiple strategy can improve can enhance the performance. We also this is some sorry. We developed personalized with multimodal gas sensors. We applied the early diagnosis of lung cancer with the convolutional neural networks and evaluation, computer metrics and ROC curves, and we got good result. And we also applied to the gastric cancers and patient and the lung cancer, it will show the what accuracy is 90%. We are preparing the submitting journals. So we applied in this research result to the early diagnosis and risk evaluation during the treatment. So, yes. Thank you for your kind attention.